in the division of rheumatology, inflammation, and immunity in Brigham and Women's Hospital, Harvard Medical School, which also is my alma mater. Vikram completed his PhD from the Division of Endocrinology at CDRI in 2014 under Dr. Ritu Trivedi, with flying colors, receiving the coveted Young Investigator Award from the International Osteoporosis Foundation. He also received several institutional awards under Best Paper category and Best Poster Award from the Indian Society for Bone Mineral Research. Very importantly, his contribution to research on Dalbergia shishu leaves in preclinical model of osteopenia uh, provided the foundation for its clinical development and subsequently its commercialization as Reunion for Rapid Fracture Healing, about which students, fellows of CDRI are all aware. After completing his PhD, Vikram moved to he moved to University of Massachusetts Amherst, where he was involved in elucidating how cranial neural crest cells migrate and contribute to craniofacial development. Upon completion of his tenure there, Vikram then joined Brigham and Women's Hospital in the Department of Orthopedic Surgery and very interestingly studied the role of one Wnt agonist LGR6, and he defined it as a novel marker for osteoprogenitor cells in bone marrow. From the division of orthopedic surgery, he made a move to this current division of rheumatology, inflammation, and immunity to study osteoarthritis, and about which he will speak today. And the title of his talk is Precision Medicine in Bone and Joint Diseases, a spatial transcriptomics approach. Vikram,
Yes,
please
sir. Yep.
begin your presentation.
Okay. So am I audible, right? And you can see my slides properly with cursor.
Yeah, very clearly. Yeah, please.
Okay. So thank you, Nevedya, for such a nice introduction. And I would like to first of all, I would like to thank CDRAST program as well as Dr. Nevedya Chattopadhyay for giving me opportunity to share my work, and which is really helpful for us getting feedback from you all. Um, so as Nevedya introduce about me, my name is Vikram Kherger, and I'm a postdoctoral fellow in Kevin Way's lab. I'm working in Brigham and Women's Hospital. So basically, our lab is mostly working on fibroblast biology, and recently our approach is like identification of various cellular targets using spatial transcriptomic approach and validating those in human organoid system. So in our lab, we are using very cutting edge techniques like spatial transcriptomics and uh, organoid system to give a putative therapeutic targets. So okay. So in today's seminar, my today's agenda is I'm gonna talk about a few slides about what I have done in my PhD studies, where I will talk about identification of bone anabolic agent, which is a part of traditional medicine. As you all know, traditional medicine is basically it involves like symptoms to diagnose the disease, and the beauty of this traditional medicine, it can be given to bulk population, but at the same time if within a population, if there is uh, it if some of the person, they are like having different composition, or let's say they behave differently to that particular medicine, which is not very well addressed in traditional medicine, then as Nevada introduced, I have work on two different projects, Adam 13 as well as a role of LGR6 in osteoblastogenesis. I will talk a few slides about that, which both involved cell-based therapy, uh, as this Adam 13 is mainly involved in cranial neural crest migration. So targeting those particular cells will be really helpful in targeting cancer, as well as useful in uh, birth defect as well as bone abnormalities. Whereas a role for LGR6, where I identify LGR6 as a osteoprogenitor marker, and so in a future, there is a possibility that because LGR6 is mainly present in mesenchymal stem cells, so if we somehow able to isolate those cells, and there are certain non-union fractures in older ages where there is no mesenchymal stem cells, so we provide a therapeutic strategy where those isolated cells can be directly injected into the patient samples and then can be fastened the fracture healing process. And in a last few slide, or in the last part, what I'm going to talk about, defining cellular target using cutting edge techniques like spatial transcriptomics and organoid system. I recently joined this lab, and I will talk in a third part about how this thing will lead to precision medicine. And precision medicine is a revolutionary approach for disease prevention and treatment that takes into account of individual differences in lifestyle, environment, and biology. This is very important, and right now people are more focusing toward that because it is very individual based therapy. So I will talk about that in a last part of my slides. So with this start, I started my PhD in identification of bone anabolic agent, where we use various different approaches. Like for identification, we after identification we use natural as well as synthetic approach, where we identified various natural molecules like vidaferrin and Alvergia ciso, which are involved in bone formation. Then, with the help of chemistry people, we also synthesize various molecules like coumarin analog, which are again involved in bone formation, and we have tested them in various animal model. We have also used a proteomic approach, where we perform a proteomics and we identify different targets by different anabolic agent, and those target can be used as a molecular approach. So where we provide with this approach, we provide a potential therapeutic agents in bone formation. For this, we use various cells like osteoblasts, chondrocytes, and adipocytes. And my work mainly focused on adipocyte cells, whereas other members of my lab, they were working on adipocytes and chondrocytes. So over there, what we did, we used in our preclinical study, we used two different models: fracture drill hole model and overectomy model, to prove that those agents are either helpful in bone formation as well as in fracture healing. As you can see, what we did over here, we perform a drill hole injury, and after performing drill hole injury, we treated those mice with various our natural and synthetic molecule, and see the time course event, how the molecules help to uh, heal this fracture healing process. This is a typical fracture healing process which require approximately day 21 to 36 in mice model. And so we check whether our particular molecule fasten then process or not, or improve bone quality uh, after fracture. We also use two other different model, where we perform overectomy and then we treated those molecule, which is known as preventive study. And after that, and in another approach, we wait for 6 to 8 weeks to develop osteopenic condition, and then we provide a treatment. And then we perform analysis using micro CT, as well as we check uh, their ability of mesenchymal stem cell differentiation towards osteoblast cells. So With this, what we identified through our preclinical study using in vitro, in vivo, and ex vivo approach, we identified various natural compound and like vidaferin A, and a synthetic compound like coumarin anabolic agents. Uh, so with this bone background, I developed a very strong interest in craniofacial development. So I joined Dr. Alfandari's group to study uh, how cranial neural crystal migrate and which is involved in craniofacial bone development. So I will talk very briefly about that project, about how ADAM13 controlled PCNS function during cranial neural crystal migration. ADAM13 is a metalloproteinase enzyme which breaks various cell surface protein. PCNS is a cadherin protein, and it belongs to cadherin family, and its full name is protocadherin, and it is present on the cell surface. This ADAM13 binds to PCNS, and it cleaves it to perform neural crystal migration. So as you can see in this is typically how CNC migration happens. This is ectoderm and this is neural tube. This neural tube, after closing, the cells migrate, as you can see in this figure, and then migrate. So this is a typical xenopus embryo, which is like fixed in a gel, and we injected RFP protein so as to track those particular cell. And as you can see, the way it is shown here in a diagram, these cells also migrate. So if you see this migration, the cell surface have various cell uh, adhesion protein like cadherins. These helps to like cell adhesion, and cleavage of this is required so that cells can move, which is also known as epithelial to mesenchymal transition. As you can see in this image, the cells migrate. If you see this representative image, there is a cleavage of particular protein because of which the cell starts migrating. And because of these two processes, as you can see, which results and which are very orchestrated process and which are required for craniofacial development. So this is how the cells from here they migrate and they perform our facial structure or facial bone. And these two process, migration of cells as well as how the cells, the cleavage of protein particular happen, and then the cell migrate, which is a very balanced and very well controlled process, and it involve it is very highly regulated process. So over there, what I did, um, I injected a morpholino. Morpholino is a molecule where, like siRNA, it used to knock down particular levels of that protein. So I use morpholino against ADAM13, and I injected it in a xenopus embryo, and I took out CNC explant and grown them in conditioned media. As you can see, those cells they are growing. And then we collected the conditioned media to see whether what are the changes in control compared to after addition of morpholino. And with LCMS, we identify a new substrate called as PCNS, which is part of cadherin family. It's known as protocadherin. So using a proteomic approach, we identified PCNS. So so after that, what we have shown is this ADAM13 cleaves PCNS to release its extracellular domain. To identify that particular PCNS protein, what we did, we cloned PCNS and we introduced flag tag so that we can detect N-terminal region, because we predict that this is the cleavage site. Basically, it has EC1, extracellular domain 1, 2, 3, 4, and this ADAM13 binds, predicted binding site is here, and it binds over here and it cleaves, which resulted in release of this particular protein, and because of which the cranial neural crystal migration happens. And to detect C-terminal domain, we generated a PCNS antibody in our lab, So that's how we are able to detect if ADAM13 is binding to PCNS and cleaving its N-terminal and C-terminal region. So we can able to detect both of this region. So what we did over here, we transfected cells with PCNS, and then we added ADAM13. E2A is a mutant of ADAM13, and we added ADAM13, which is a wide form. And what we have seen using Western, that there is EC124 domain. As you can see, we are able to detect this particular region. And so PCNS, and this is the control where we have seen that PCN is transfected very well, as well as ADAM13 is also transfected. So which shows that ADAM13 binds to this particular region and it cleaves PCNS molecule and releases its extracellular domain. So this is a representative image of where ADAM30 binds and it resulted in release of EC124. Now it releases EC124, but whether it's functionally important. Uh, so to answer this, what we the next experiment we performed, we use Morpholino. We injected at one cell stage for ADAM13 for knocking down of ADAM13, and then we injected RNA at 8 cell stage. Why we use 8 cell stage? Because this particular stage at this cell give rise to uh, cranial neural cells. So that's why we particularly injected those cells. And as you can see, this image, this is a representative image where we use counting how this migration happens. So these are the representative images after injection. As you can see, RFP ADAM13 morpholino, there is no migration of cranial neural cells, whereas RFP show a very good migration, which shows that ADAM13 blocks migration of or remove knocking down of ADAM13 blocks the migration of cranial neural cell. Whereas when we externally injected EC124, it show a very good migration compared to ADAM13 uh, morpholino group. Whereas when we added the whole PCNS molecule, it also rescued. And ADAM13, which is we considered as a positive control, it completely rescued. So compared to ADAM13, those the rescue is little bit less, but still they are comparable to that of RFP. So from this, we can say that inhibition of CNC migration by ADAM13 morpholino was rescued by PCNS and its cleaved fragment EC124. So from this, we can conclude that ADAM13 regulate PCNS function via AP2 alpha. I have not shown detailed data for AP2, but what we observe is that AP2 alpha is a transcription factor. This ADAM13 cytoplasmic domain somehow regulate AP2 alpha expression, and this AP2 alpha guides the PCNS expression. So I haven't touched that data, but we have already shown in our paper. And there is another function of ADAM13 is that ADAM13 binds to PCNS, which cleaves EC124 fragment, and which is a part of extracellular domain. And these two processes, the cytoplasmic domain, which resulted in AP2 alpha expression, and it guides the PCNS expression, as well as the cleavage part, resulted in a CNC migration. So the first part is cleavage driven regulation, where ADAM13 binds and it cleaves EC124, and because of that, the cell migrates. And another is there is AP2 alpha and PCNS expression axis, which also directly regulate uh, CNC migration. So this study shows that metalloprotease can promote cell migration in embryo by cleaving and regulating multiple genes. Then they may also promote cancer cell expression. And understanding how metalloprotease work and how we can stop their function may also lead to various new approaches to the birth defects, bone deformities, because mainly these cells involved in facial bone formation. So various facial deformities can be easily, or I will not say easily, but can be understanding this particular mechanism will help to provide a new therapeutic approach. as well as because there is EMT transition is happening. So this is a really good strategy for cancer treatment too. So while working on cranial neural crystal, I came across this paper, which is known as, like role of LGR6 that marks nail stem cells, and it is required for digit tip regeneration, which strikes me and which is very interesting. Why it is interesting? Because during digit, so basically mouse digit tip, if you cut mouse digit tip, it regenerate again. So uh, when it gets regenerate, like as you can see, this is a sequential process of mouse digit regeneration. uh, in this paper, Jessica saw expression of LGR6. LGR6 is an agonist of Wnt. So uh, when I was reading that paper, it was very interesting. If there is bone formation happening and there is LGR6 is very expressed around that particular regenerated bone, that means that there must be LGR6 may be performing some function in bone formation or bone regeneration. So, and that this LGR6 may be helpful in remodeling of bone or maybe repair. So that is a really good uh, therapeutic target for fracture healing process. So, and which can be used as a cell-based therapy. So that's why it interests me. So I started to look at the role of LGR6 in osteoblastogenesis. So in that paper, uh, author shown is that this is a digit tip section where after amputation, you can see there is SP7, which is a marker for osteoblast cells, and these SP7 positive cells were co-localized with LGR6 positive cells. So this is basically a lineage tracing experiment where the mice were breeded with, along with the Rosa mice were bred with LGR6 3 mice. So all the cells which are expressing LGR6 will be shown red in color. So as you can see, this SP7 and tomato cells, they co-localize, which suggests that SP7 positive cells are LGR6 positive cells. And in that paper, they have shown that when there is removal of LGR6, the mouse digit tip regeneration is hindered, as you can see, compared to this. Uh, the knockdown or the knockout of LGR6 resulted in very hindered digit tip regeneration. And as you can see, this is a percent regeneration graph where compared to wild type, heterozygous doesn't show much change, whereas LGR6 null mice show very strong change in percent regeneration. So from that, so these are the two very interesting clue. And then secondly, when I was looking at other bone, so the author showed that LGR6 express in mouse digitive, whereas when because uh, the section contains other long bone too, so I started to look at those section, and what I observe is LGR6 is basically present on the long bone which is adjacent to digit. So which definitely give a very strong clue that this LGR6 involved in, or it is doing something in um, bone formation or bone regeneration. So what I did after that, I took bone marrow cells and calvaria, and I checked LGR4 and LGR6 expression, and I keep skin as a positive control. Why I use LGR4 over here? Because LGR4 is pretty much 65% similar to LGR6, and lots of studies have done on LGR6. So it's a kind of like a control to see how much LGR6 expression is different than LGR4. So what I observed that compared to bone and bone marrow cell, calvaria show a very strong expression of LGR6, and this is comparative to skin cells. So 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 that's why we think that the LGR6 may have broad role in osteoblastogenesis. So a little bit background about LGR6 in Wnt signaling. LGR6 is a leucine-rich repeat G protein coupled receptor, It is a agonist of wind signaling, and wind signal play an important role in bone development. So, and this is the signaling pathway of LGR6, where you can see uh, when there is uh, when there is no R-spondin, which is a ligand for LGR6, this receptor degradation happens, and then there is attenuation of wind signaling. Whereas when R-spondin binds to ZNFR, this which resulted in enhanced wind signaling because of this ZNF ubiquitination. So, and again, this R-spondin and MARI, which are known to bind to LGR6, and R-spondins are necessary for bone development. Uh, and MARI, which is a ligand for this LGR6 receptor, it is also uh, known to improve fracture healing. So, and the recent paper shows that LGR6 also play a role in osteoblastogenesis by activating mean signaling in MC383 even cells. So, all this literature and whatever preliminary data what we have seen uh, give a very strong proves that this wind agonist LGR6 may involve in bone formation. So our next question arises, when is LGR6 expressed? To answer this question, what we did, we performed a mesenchymal stem cell culture, and we grown those cells in osteogenic media, and we collected RNA at different time point. And as you can see in this experiment, after collection of RNA, we check the expression of LGR6 at different time point, like time 0, 1, 3, 7, and we culture those cells till day 21. And what we have seen is LGR6 are very strongly induced around day 1, and then its expression goes down very drastically around day 3, 7, 11, 14, and 18. And we have seen this data similarly with mm, Western blot, so which confirms basically our results. And what this data shows that LGR6 is expressed at all stages of osteodifferentiation, and LGR6 expression is sharply increased upon osteoinduction, as you can see over here, and then it decreases the remainder of the process. Like after day 3, the levels remain very low, but it's still expressed at all stages. And we have also checked in other cell line, not cell line, I will say we checked with other primary cells like bone marrow cells as well as calvarial cells. So the next question is, where is LGR6 expressed? To answer this question, what we did, we took bone, this is femur bone, and we checked and we use LGR6 antibody, and we perform immunohistochemistry. And what we have seen is this LGR6 positive cells are present at the border of trabeculae, which is very interesting. And because on a trabecular on the surface, there are two types of cells, mainly osteoblast and osteoclast. So we co-label this LGR6 positive cells using TRAP and SP7. And what we have seen, uh, this LGR6 positive cells are distinct from TRAP positive cells, whereas they localizes with SP7. So this shows that this LGR6 is LGR6 expressing cells are osteoblastic cells, or they are pre-osteoblastic cells. So then the question is LGR6 is necessary for osteoblastogenesis? To answer that particular question, what we did, we took mesenchymal stem cell as well as osteoblast cells, and we grown them in osteogenic media. And then we added siRNA against LGR6, as well as we use LGR6 plasmid. And then we check the effect of osteoblastogenesis using various marker SP7, RUNX2, alkaline phosphatase activity, and mineralization. And what we observed that uh, after 24 hours of transfection with LGR6 siRNA, there is almost 50% reduction of RUNX2 level, whereas after 72 hours, the reduction reduces a little bit. Whereas in case of SP7, the reduction is even more after 72 hours. which suggests that knocking down of LGR6 reduces osteogenic marker, SP7 and NX2. Then we have checked alkaline phosphatase activity, which alkaline phosphatase activity is a marker for osteoblast differentiation. As cells become committed pre-osteoblast, at that point, alkaline phosphatase increases, which is a cell surface marker. So, what we have done over here, we transfected cells with siRNA against LGR6, and then we we check the alkaline phosphatase activity after day 1, day 3, and day 7. On day 1 and day 3, we didn't observe like much changes, but around day 7, we saw almost like a 20 to 30% reduction in alkaline phosphatase activity. After that, we check mineralization after 21 days, and what we observe is with LGR6 SIRN or knocking down of LGR6, there is reduction of um, uh, alizarin red staining, which is a marker for mineralization. So we shows that there is reduction of mineralization. So after that, this is we use siRNA approach where we reduce LGR6 expression or we knock down LGR6 expression. But now we use gain of function using plasmid. So we transfected plasmid because ideally, if LGR6 removal of LGR6 reduces those functions, so the next question is whether overexpression increases those particular marker. And what we have observed is after transfection with LGR6 or overexpressing LGR6, alkaline phosphatase activity and osteopontin increases, whereas we have not seen much effect on NX2 and SP7. This is day 7, so that's why probably we have not seen much effect on NX2 and SP7. Then we have also checked alkaline phosphatase activity around day 1, day 3, and day 7 after LGR6 overexpression. And we have seen increase in alkaline phosphatase activity around day 7. And similarly, we have also seen increase in mineralization. So then a very next obvious question is, because we have seen LGR6 is important in cells, so whether it is necessary for bone maintenance. So to answer this question, what we did, we took LGR6 wild type and null mice, which are 8-week old, and then we took out femur bone and we performed a bone parameter analysis. At the same time, we also took bone and we flush bone marrow cells, and then we culture those cells in osteogenic media to see ability of the bone marrow from wild type as well as LGR6 null mice toward differentiation, towards osteoblast differentiation. So what we have seen is deletion of LGR6 decreases various trabecular bone parameters. So as you can see, the blue is male and red is female. So with LGR6, removal of LGR6, male as well as female both show reduction in BV by TV, whereas trabecular separation was increased in male with reduction of, or say, removal of LGR6, whereas female mice don't show a much strong effect or they are not significant. And when we check trabecular thickness, we haven't seen much effect on male, whereas female show slight reduction, which is significant. Similarly, we check trabecular number where we have seen effect on male. SMI was significant in female, whereas connective density was significantly reduced in both male as well as female. Then after that, we perform ex vivo culture and we check the alkaline phosphatase activity. And we took bone marrow cells from LGR6 wild type as well as LGR6 null mice, and we culture those cells in osteogenic media. And what we have seen, removal of LGR6 resulted in reduced or reduction in ability of bone marrow cells to differentiate toward osteoblasts, which is evident by alkaline phosphatase activity. As you can see, there is reduction of alkaline phosphatase in male as well as female. Similarly, this result was also corroborated with mineralization, where we have seen there is a strong decrease in mineralization in male compared to as well as in female. So from this, we conclude that LGR6 show a dynamic expression in osteoprogenitors. LGR6 expressing cells are present on the surface of trabeculae, and those are osteoblastic cells. And LGR6 play a very important role in differentiation of osteoblast cells, uh, as we have confirmed using siRNA as well as plasmid approach. And LGR6 is genetically necessary to attain peak bone mass. So this is our working model where we have shown that when mesenchymal stem cell converted into pre-osteoblast, there is a very strong induction of LGR6. Uh, and this LGR6 may lead to induction of SP7 or RUNX2 directly, or maybe some intermediate protein involved, which resulted in differentiation and mineralization, and eventually osteoblastogenesis, and which may involve in bone homeostasis or bone formation. I haven't touched the ligand part over here, but what we observe is that MARI, which is a ligand for LGR6, it binds to LGR6, and it mainly involved in proliferation of the cells. And it don't have much stronger effect on differentiation. Whereas R-spondin, there are four R-spondin which are ligand for LGR6, 1, 2, 3, 4. And what we observe, R-spondin 2 is, is mainly involved in differentiation, whereas it don't have very strong influence on proliferation. So what we believe, basically, this initial part was played by MARI, whereas the differentiation part was played by R-spondin. So this LGR6 is kind of act as a switch, and which involves like which ligand binds to that particular receptor and perform its function. So with this background, as a bone biologist, I developed a very strong interest in osteoarthritis and joined Kevin Wei's lab, which is very well established in bone fibroblast biology and arthritis research using cutting edge like single cell sequencing and spatial transcriptomics. In this lab, I started working on defining cellular targets in osteoarthritis. So a little bit brief introduction about what is osteoarthritis. So acute direct trauma causes the onset of synovitis, and it increases risk of osteoarthritis affecting millions of individuals. And the possibility of development of OA within 3 to 10 years is even more when you have any trauma. And then and there is no medical treatment available to prevent OA after injury. People have gone for like tradition. People go for traditional medicine or exercise, but the ultimate therapy is surgery. So which is very maybe very painful. So and identification of cellular mediators of post-injury synovitis hold a very strong promise for developing a therapeutic strategy to halt inflammation and prevent early onset of OA. This is a representative image of how osteoarthritis progresses. This is a normal knee joint or normal knee image. As you can see, this is a healthy endochondral unit which consists of chondrocyte, articular cartilage, cartilaginous matrix, and calcified cartilage, and this is cortical plate. And when this is the early progression of OA, maybe after injury, and some of the reports suggest that even the injury is not even important. Maybe eventually with older ages, people may develop OA. So this shows that there is a chondrocyte metabolic activity, the release of NOROS, and which resulted in increase in pro-inflammatory cytokines, which resulted in a development of surface fibrillation. As you can see over here, there is water content and swelling and cortical bone porosity, and eventually this process, if remain untreated, it increase the inflammation increases, inflammatory cytokine increases. As you can see, the subchondral bone mass and alteration of there is alteration of architecture happen, the cortical thickness changes, and there is fissuring and fragmentation, and there is osteophyte formation, and there is also both formation, and which resulted in established OA, which is a late stage OA, where you can see clusters of chondrocyte, there is apoptotic osteocyte, there is osteophyte formation, and sometimes even like fusion of bone also happen, which makes a very painful condition. So yeah, so that's this is the background about osteoarthritis, and our lab is basically working on rheumatoid arthritis as well as osteoarthritis, and I'm primarily focusing on osteoarthritis. So this is basic background of our lab, where our strategy is we, in this lab we are mainly working on human tissue samples. So what we do is like we take synovial biopsy samples, and we dissociate those synovial tissue samples using enzymatic process like liberate and dispase, and then we perform single cell RNA sequencing, which is known as disease reconstruction, to identify various novel pathogenic cells. And then uh, this will define the heterogeneity, what different type of markers are expressing, what different types of cells are expressing, and which will be helpful in identifying biomarker, and that can use to predict a treatment response. Also, after that, we also use spatial transcriptomics approach, where we take a synovial biopsy samples, we perform uh, spatial transcriptomics on those tissue samples to construct the tissue pathology, to see what uh, what's localization of that particular inflammatory cell, as well as the interaction of different cell, how immune cell interacting with fibroblasts, and that can be used as a therapeutic target. And then we reconstruct, or this part is like known as disease reconstruction, where we use patient derived organoid. So this synovial biopsy samples, we grown those in media, and then we uh, check the response by using various different techniques, and that can be used for precision medicine. So basically the strategy is like taking synovial biopsy samples, uh, and then perform a single cell RNA sequencing, at the same time perform spatial transcriptomics so as to identify various types of cells, and then use a particular patient-derived organoid and study what type of chemokines and chemokines are secreted, or what cell types are there, and that can help to guide or help in precision medicine approach. So this is, as I have shown you. So basically, we take a synovial biopsy sample and we perform single cell RNA sequencing. I'm gonna give a very brief introduction about single cell RNA sequencing, where there is like 10x barcoded gel beads, and then the cells and nuclei along with enzymes were injected, then they are collected, and then this barcoded cells were the libraries were generated, and those library were sequencing was performed, and then after performing the sequencing, and it is used for analysis, which resulted in so basically this uh, collection of cells will after analysis will show which different types of cells are present in that particular synovial sample. So this is my experimental approach. As I have shown you earlier slide, the single cell RNA sequencing approach. Here, this is synovial sample from OA patient, where we perform single cell RNA sequencing by dissociating the synovial tissue samples, and what we observe, there is a very heterogeneous cell population. We observe fibroblast cells in that particular patient sample. We saw myeloid cells, CD4 T cells, CD8 cells, and natural killer 2 cells, and NK cells. So we are right now characterizing those cells, um, and we are trying to, so there are different patient samples at different time points, so we're gonna look at the changes in different pathological cells, so that can be used for precision medicine. So then next step is spatial transcriptomics approach. This is a basic workflow of cosmic chemistry and workflow, where you can see uh, this is tissue. So basically, you take a synovial biopsy samples, then you made a slide. Uh, the the RNA is exposed in a tissue, then the IHC probe is added for the hybridization, and there is cyclic readout using various reporter, so that so you can particularly identify the location of the cells or which protein is expressed. So this is a workflow how it is done. So analysis of workflow to transform raw images to decoded RNA transcript at single cell subcellular resolution. So these are the raw images where after performing histology, so there is a morphology based say segmentation. Then there is segmentation is done using various markers, and then after the cell segmentation, various proteins are identified on those particular cells. So for segmentation or subcellular resolution, there are various genes like mitochondrial genes, cytoplasmic genes, and nuclear genes are used for localization or identification or checking the exact location of the cells. And the third is this table shows the quantification of transcript detected in 3-cell compartment. So this is basic design of spatial transcriptomics. And so this is again what we are right now working on. This is against synovial sample of OA patient, where we perform a spatial transcriptomics using cosmic spatial molecular imager. And what you can see, we are able to identify various types of cells in that particular tissue section. This is a large image, and this is single cell perform on those particular cells, and this is uh, particular segmented cell on that tissue. So we identify various cells like CD8 cell, lining fibroblast, myeloid cells. So this orange color shows say CD8 cells. So these are present in that particular region. So around that, what type of cells are present, how these cells are interacting to each other. Right now, we are working on that. At the same time, on after identification of cells, we would like to know what different markers or what different genes are uh, increasing over here, or what different markers expression is happening, or there is a changes of ligand or receptor. So that can be analyzed using this spatial transcriptomics approach. And it is because single cell will not allow you exact localization of the cell, whereas spatial transcriptomics give the exact localization as well as which neighboring cells are present around that particular cluster, which is a very helpful to identify various pathological cell as well as fibrotic or pathological niche over there. So after performing the spatial transcriptomics approach, my next approach, how to validate. So as you can see, we use over here a patient-derived organoid system. Here we characterize organoid by taking that particular sample, synovial tissue samples, and grown them in matrigel and track that particular organoid for 21 days. As you can see, around day 9, the cells started to come out. Maybe the image is like little bit smaller, but in a surrounding, the cells come out. At the same time, the organoid system shrinks a little bit, which is show that it is growing well. And then we perform histological section to see uh, inflammatory cells at different day times, so as we can validate whatever therapeutic targets we saw using special transcriptomics or single cell RNA sequencing that can be validated in organoid system. And we have also, this is a CD31, a CD30, we use CD31 antibody to see the vascular network. So the rationale behind this is like, as you can see, a very well perform a well vascular network is here. So if if you add particular compound, or if you see there is a changes in vascular network, that also can be monitored by organoid system. Um, so direct validating approach is like, we are able to identify various cytokines in patient-derived organoid from synovial tissue, as you can see, by using O-link platform. So and also a tofacitinib, which is a inhibitor for which is used as a treatment in arthritis. It shows a reduction of IL-6. So basically, the strategy is like, if we saw some of the marker or compound, or any particular protein is increasing over here, so basically taking that particular patient derived samples and see how this particular drug is responding uh, to that organoid, which is really helpful in guiding precision medicine. So with this all background, so cellular profiling techniques such as single cell transcriptomics and spatial transcriptomics will help to uncover novel pathogenic cell type. Special transcriptomic approach will help to see changes in cell-cell interaction that retain each patient characteristic. And this patient-derived organoid system is very promising approach that may provide a platform to validate therapeutic targets and determine the therapeutic response in existing therapy. So together, this insight provide a very exciting opportunity to translate discoveries into precision medicine. So back to my original slide where I have worked on traditional medicine and cell-based medicine. So right now I'm working on more on precision medicine, and this result from our study will help to provide a foundation to target inflammatory cell, as we are able to identify various CD8 positive as well as CD4 T cells, so that inflammatory cells uh, can be used as a target, and their mediators, which are like any mediator which are increasing the population of particular cell type, can be used as a therapeutic potential therapeutic targets, and that contribute to inflammation and progression of OA. So acknowledgement, I would like to acknowledge my PhD group, my first postdoc group, as well as my second. At the same time, I would like to acknowledge my current lab member. Thank you. Questions and comments?
Okay, excellent. You have very nicely and coherently taken us through your research career, so to say. So from the beginning till this point, and particularly the background for spatial transcriptomics was very helpful. I'm sure that I personally have benefited a lot from the way you have described and made us understand its importance and how it is done. It is indeed very exciting, I must say. And also your work on cell-based therapy in your previous lab, where you found that LGR6 has the potential to be a early marker, osteoprogenitor marker in the bone marrow. Now, a little bit, I don't know much about LGR6. It is basically a G protein coupled receptor, right? So, how does it
Yeah.
really
Yeah.
modulate Wnt signaling or Wnt pathway?
So, like Wnt pathway, as I've shown you, there is like a ZNFR ubiquitinating protein. So basically, LGR6 is present very, like say, distinct from that particular cluster. And when there is ZNFR is not bind or it is not ubiquitin related, it is the Wnt signal is inactive. But when the R spondin binds to LGR6, uh, it resulted in degradation of that ubiquitination of ZNFR, ZNFR3, which resulted in activation of signaling. And that signaling involved, so if there is, so basically what we have seen, if there is MARI, which is a ligand for LGR6, if that is acting, that axis is acting, that it works via CMP pathway. Whereas when R spondin activate, it has a different like it goes like various involvement of osteogenic gene or increase in osteogenic gene. So that's, I think I answered
I
your
see.
question, or
Yeah. Okay.
yeah.
Yes, yes, very much. And another clarification regarding your most recent work. So you
Mhm.
you are taking for organoid culture, you are taking, it seems, patient samples. So are you harvesting the FLS from there to begin your organoid culture?
Yeah, so basically what we did, so whenever there is operation goes, and so we go there and there is discarded tissue, synovial discard, people go for knee surgery, or there is ACL surgery. So
Right.
there is lots of discarded synovial tissue. So we collect those particular tissue and then we culture that thing. So basically we
No,
have,
I mean,
yeah, sorry.
yeah. So are you basically taking or getting FLS from there to harvest for your culture, or you
Yeah, yeah,
you
yeah.
uh, you take a mixed bag of cells?
So basically, we also get a mixed bag at the same time. Some of the surgeon they give us like a very specific FLS related tissue, and some of it's because it depends upon what kind of study we are doing. So if we are like more focusing on FLS, we will use those particular FLS oriented tissue samples, whereas uh, some of the surgeons they gave us like a whole chunk of like tissue sections. Then we take that tissue, we come to our lab, and then we dissect that tissue which is for our particular interest.
Hmm, okay. And then finally, about LGR6, is there any facts possible with, for example, if you have to check it in the bone marrow, can you do a
Mhm.
fact-based sorting for this kind of cells?
We tried a lot doing, the only thing is like antibodies are not really good at that point. We tried. There is one APCAM antibody, but it requires fixation because LGR6 is a cell
Oh,
surface protein, and the maximum
okay.
antibodies were like on a cytoplasmic domain. So it's like a very tricky thing. You can do it, but at the same time, you may lose certain features.
I see. So it is not really reported or has been optimized for fact sorting kind of a thing, has not been optimized yet.
Not yet, but I will
You
definitely.
need fax grade antibody, which I mean, is not available, right?
Yeah, I did it using fixed tissue because the
Mm,
particular antibody which is we
yeah.
ordered like 3 4 antibodies and we tried with that. Only one
Mm.
antibody worked, but it required fixation and all those process.
Oh, okay.
Like
Okay.
processing is very high.
Right. Yeah. Okay. My question is, I'm done with my question. If any other question, please go ahead. I unfortunately I don't see. This is very very funny. Why I don't see anybody from your previous lab? Anyway, they will be see. They will see your video of it. Okay, if there are no other questions, then let me thank Vikram again, and really enjoyed your talk, and it is a very exciting work you are doing, and keep up your good work, and all the best.
Yeah. Right now it's like a very preliminary, but I think within like because we ordered a special transcriptomics instrument, which is gonna come, which is due coming on this February. So most probably, yeah, everything is like this
Yeah,
year gonna be
sure.
crucial. Yeah, yeah. And I
Yeah,
started like last 6 months, so pretty like new for me too, but trying to understand it. Hope so you like and um, what I'm doing. It's like really helpful over there.
sure, sure. Okay, excellent, and all the best. Thank you
Thank you.
very
Thank
much, Vikram.
you.
Thank you.